Pathogens very commonly use cells as host sites for their replication. This is done via endocytosis and has had devastating effects on people and livestock. So why is this done? Well, viruses have no other option. They must insert their genomes into other cells in order to replicate their DNA using the host machinery. Non-enveloped viruses utilize endocytosis involving the intercellular membranes of the host cell, whereas in enveloped animal viruses, the membranes act as a vesicle to transport the virus. Bacteria use endocytosis to utilize protected environment of the host cell's cytosol. The bacteria secrete proteins such as toxins to bypass cellular defenses. Proteins that are secreted may either allow modification or trigger escape from vacuoles, which can then recruit actin to move through the cell. Secretion systems are also used to transport proteins from the bacteria to the interior of the host cell. The proteins often mimic eukaryotic proteins and interfere with cellular mechanisms. As a part of the endocytic process, cells trigger a pH drop that many viruses in turn use as a cue to activate penetration. Viruses can either fuse their envelopes with the organelle membranes to avoid exposing their detectable glycoproteins, or they can release their capsids into the cytosol. The diagram to the right summarizes the endocytic process, listing relevant pHs to the left. Clathrin-mediated endocytosis is the most common mechanism for endocytosis of small viruses. Viruses can induce clathrin assembly at the site of their binding, and after pit formation, the coated vesicles pinch off with the viruses. The coat then dissociates and the viruses are taken to the early endosomes. Vesicular stomatitis virus uses CME for productive infection. The clathrin coat is recruited to binding sites for VSV, but because of its bullet shape and large size, the clathrin vesicles form slowly, with some even only managing a partial coat. Viruses like VSV can induce CME locally and adapt it to their needs. Clathrin and caveolin independent pathways exist also for some viruses, like SV40. By binding multiple GM1 molecules, SV40 generates membrane curvature and appears as though the plasma membrane is wrapping itself around the virus particle. The image shows human papillomavirus 16 entering HeLa cells via a mechanism similar to macropinocytosis, where a filopodia is bound and the endocytic process is activated independently. Caveolar endocytosis is a proposed method of viral endocytosis, but is difficult to determine as the endocytic role of caveolae in cell life is poorly researched. SV40 is the most studied of the viruses that may partake in caveolar uptake. Morphological evidence shows colocalization of the viruses and caveolin-1 in plasma membrane pits. The addition of SV40 to cells causes dramatic tyrosine phosphorylation-dependent elevation in a caveolar vesicle formation and motility. Depletion of caveolin-1 has shown to result in a decrease in SV40 endocytosis and infection. Additionally, depletion of a caveolar ATPase was shown to cause an increase in caveolar vesicle trafficking and efficiency of the affection. Thus, SV40 may utilize caveolin-independent and dependent pathways. The rate and efficiency of entry of bacteria into cells vary greatly with the cell type, temperature, or growth phase. Main components of the zipper mechanism are a dedicated bacterial surface protein or component that interacts directly with the host cell receptor to induce a series of signaling events that culminates in endocytosis. The actin cytoskeleton membrane composition and its plasticity are critical elements as well. Listeria monocytogenes is one bacterium entering by the zipper mechanism. It expresses two proteins involved in entry. Interalin A is a surface protein that interacts with E-cadherin expressed only in some epithelial cells. Interalin B proteins activate the receptor tyrosine kinase MET, the hepatocyte growth factor receptor that is expressed on all cells of epithelial origin. Electron microscope analysis of cells infected with listeria show the presence of clathrin on membranous invaginations as isolated clathrin-coated pits. Shigella and Salmonella are two bacteria that use trigger mechanism to enter cells. This mechanism is defined by the formation of large actin-rich membrane ruffles triggered by a signaling cascade of T3SS that induces localized transient changes in actin dynamics. These are just a few ways viruses and bacteria can enter a host cell. This research is important because a better understanding of the endocytic pathways in host cells can provide new options in the development of antiviral and antibacterial strategies. Pathogens can also be valuable tools in biological research. They have been used to study membrane biology, signal transduction, actin-based motility, and more. By studying them, there becomes the possibility to develop quantitative assays with the infection itself and to follow the fate of a single pathogen in live cells using light microscopy at increasingly high resolution. The link below leads to the NCBI article that was used to collect information for this presentation.